So since we're at the policy meeting, I decided to adhere strictly to the policy guideline on how the slides should be formatted. But since we're about to change policy, I also made some ad lib modifications. So the first three slides, there's only 6 slides total, but the first 3 slides will be a little bit not that much about policy, but about the framework that is changing around us that then induces changes in policy. And just to give a sense of where we are today with the genomics and actually more generally with the omics having done a tremendous, very much thanks to improved engineering, a terrific job of cataloging the parts. We're now in a position where if cancer were a broken Swiss clock, an incredibly complicated broken Swiss clock, this is what we would look at in an extraordinarily complex set of individual gears. And we would try to figure out what is broken by looking at each gear and finding out whether it is broken in multiple clocks that display the same symptoms of brokenness. And I think the way systems biology is coming around is by trying to figure out how things work together, essentially, and not doing so in the way that has been really the landmark science of sort of mechanistic discovery, where you look at very small aggregate of proteins or regulatory genes that control the behavior of the cell, but really looking in genome-wide fashion at the entire regulatory landscape of a cell. And in fact, realizing very quickly that there's no such thing as regulation in a human cells because every individual, and in fact every cell within an individual has a different mechanism. So each cell is a different watch. And so to some extent, what is different today in the way systems biology and other type of sort of model-driven sciences are thinking about the problem compared to what we've been doing for the last 20 years since the omics started to play a major role, is that instead of looking at traditional approaches to precision medicines where predictions are based on statistical association, today it's cloudy, then tomorrow it's going to rain, or gene X is mutated, therefore drug Y will work. We're starting to go towards a more of a model-driven type of science where essentially we start to look at creating physical regulatory models of the cell and then making prediction based on their global behavior. And I wanted to just mention that this has been probably the most phenomenal sort of accelerator of all the other natural sciences that we think of. If you look at physics, people used to roll balls down an incline and measure how long it would take until Newton came up with some Newton laws. And in meteorology, for instance, we used to make prediction based on statistics, and today we make prediction based on very complex Navier-Stokes models of fluid dynamics that allow us to understand weather up to about a week ahead. And so what we are starting to really realize with the combination of traditional genomics and systems biology is that we can use cancer cell as models to predict activity of the drug that are actually very open box. They're not black box model or white box model, where you put in some data and at the other end you get a result, but rather these are completely transparent boxes in which when the data goes in, you understand why a particular result comes out the other end. And I think that this requires really fundamental changes, not just in policy, but also in focus, because I think we need to really gather the entire community and transitioning from sort of an effort to map human genomes to an effort to map the human disease regulation model. So let me give you an example of how this works. This is obviously going to be 50,000 feet level, much as Bill has done for the other part. But this is essentially a paper that is now in review. We actually got really great reviews. Hopefully you'll see this soon. And they were very thoughtful reviews, actually, so thanks to the reviewers if they're in the audience. But for really incredibly thoughtful this time. But the idea is that if you look at prostate cancer, which is not always the case, so the model of prostate cancer, in terms of understanding it from a predictive therapy perspective, starts in many ways with an outline of all the things that have gone wrong in large cohort of patients, including genes that are mutated, amplified, deleted, genes that are dysregulated without, we don't know exactly why, for instance by gene expression analysis or phosphoproteomic analysis. But from that data, what one can do is to start creating models of regulation. When you create model of regulation, you can link individual proteins that are important in prostate cancer with a set of genes that they regulate. And sometimes we realize that the action is not around a particular protein, but is actually in the middle ground that links two different proteins together, because what they co-regulate is starting to become much more important than what each individual protein will actually regulate. And this is seen here. Think of red as being related to very aggressive prostate cancer, and blue being related to just run-of-the-mill prostate cancer. And you'll see that, for instance, if you take these two proteins, their individual targets are all over the place. They're blue and red. Individual targets are blue and red. But if you actually take their co-regulated targets, they're exquisitely red. And this indicates that, in fact, or suggests that this protein may work synergistically. This is something I want to make sure that everybody understands. Synergy and epistasis, two fundamental processes in regulation of our cells, cannot be really understood with traditional statistical approaches unless you had cohorts of incredible size. And in fact, the thing that we normally discover using genomics are individual gene contribution to disease. But in this case, what is really extraordinary is that if you actually silence one gene or the other, these tumors continue to grow. This is in the mouse PDX model, albeit at a slower pace. But if you silence both of them, you go dramatically, these tumors completely tank. And in fact, if you go in populations, you can see that the individuals that are negative for both of these genes will never die. This is over 20 years about. Will never die of prostate cancer, while the patients that have activation of both genes will actually have the absolute worst prognosis. And in fact, this pretty much recapitulates, this is an all-comer study on about 1,000 TMAs. This pretty much recapitulates the full burden in terms of mortality of the disease. Again, no statistical significance of the individual genes, so you would never have been able to detect them by individually, one at a time. Okay? And this is, I think, is where systems biology brings you perspective, which then leads to identifying drugs. In this particular case, you can find two small molecules, one inhibiting one inhibiting the other, and you can see that the synergistic effect is dramatic, 9-fold tumor reduction compared to standard of care. So this type of paradigm is a shift in the way we think about cancer. Why? Because this can be done right now in about 3 months end-to-end for an individual patient. Okay? And we have already started a series of studies we call N of 1, that complement the typical genomic studies where we focus more on mutations. Neither one of these two genes that I showed you is actually mutated pretty much ever in a cancer, prostate cancer patient, but they constitute incredible, what we call non-oncogene dependencies of the cells. That is, you shut them down, and these cells of the aggressive tumors will actually die. So why is this important from a policy perspective? So, and this is where we started to go back to the sort of meeting goals. So today, the classical study model for clinical trials is a indication therapy type of model, where you may have a particular type of breast cancer, HER2-positive breast cancer, and then in that case, one would do a clinical trial to study Herceptin. And if you're in glioma, for instance, you can have just a generic approach that says you have glioblastoma, therefore you will get radiation and temozolomide. And this is a little bit like saying if you're in Seattle, you better have an umbrella, which is okay in some of the cases, but not most of the cases. A little bit more defined, as we're getting from genomic data, is if you have a glioma with the FGFR TAC fusion event, then you may actually want to get an FGFR inhibitor. This is a little bit like saying if it's sunny today, so we're basing it on actual observation of today rather than on global statistics, you'll get suntan lotion tomorrow, but what if it rains? Okay? And I think what we're starting to do, and I think I'm incredibly excited about, Jim will hopefully talk about this later on, about this sort of match study that the NCI is pioneering, as well as bucket studies, is the fact that we will have to fragment cancer further. We're kind of out of the big bullets like HER2, and we're starting to get into more fragmented sort of cancer therapies. And if we want to win the battle and not having to design one drug for every little fragment of cancer, we'll need to figure out what are the generalizable principles that allow us to think about it and reuse the same type of drugs or drug combination. And so I want to mention there's about 1,500 distinct FDA-approved drugs and more than 1 million true drug combination. About 10% of these do completely different things than the individual drugs. So we have a repertoire of about 100,000 drug combinations that do completely novel things, which we have not characterized. And while we think of precision cancer medicine is a place where the actual model predicts which drug or drug combination we should actually administer to a particular patient. And in some cases, not just drugs. It could be radiotherapy combined with drugs, et cetera. And some of those papers are starting to come out. And so I think that the real value here and the way we have to think about policy changes is around how we design and think about clinical studies, about being not just about the indication therapy, but in fact multiple and possibly a continuum of indication therapy models that are actually driven by sophisticated models, very much like you would do it in physics if you have to figure out how certain things work. And so just to make a point that is really critical, I think, at this point in time, there are about 500 or more phase 3 clinical studies in the US today open, okay? This is an incredibly missed opportunity because the real problem is that we don't know yet whether this kind of principle of precision medicine actually work or don't work. We know that at some time they will work, but we need to know whether they work, at which points they will. And so the idea is that we need to make this correlative data from clinical trials available to the research community and allow blind prospective determination of which one will be the responder versus non-responder. This is not the way clinical trials are run today. I hope this is the way, and I'm pretty optimistic that this is the way the MATCH trial will run. And there's a proven methodology in terms of the DREAMS, a conference we've been running for the last 7 years, where we provide the community with data. We ask them to make predictions about, for instance, breast cancer prognosis, which was recently published in Science Translational Medicine, drug sensitivity challenge, and drug combination challenge that are then addressed by the community with a large number of groups participating and making predictions. And so let me finish with my policy slide, which is basically, sort of in the basic science, I think there's a dramatic focus shift that is needed. We need to move from just building omics collections to building models of regulation. And I think this is something that the community needs to really gather around, much as we've come around on the Genome Project. In terms of clinical study design, I think we have to move somewhat away from an outdated biomarker drug model and move into more of a sort of predictive model that determines what the optimal strategy is. We need to change the way early patients have access to experimental therapeutics so that we can actually put the patient on the right drugs that are predicted by these models. And then I think we need to really modify the way we think about data access policies on going from retrospective to prospective clinical data and controlling early access to clinical study data so that the entire community can be engaged on predicting where we go next. So I'll stop here.
Thank you, Andrea. We have one or two questions for Andrea at the end.
That was absolutely great. I love all your points. So thinking about, how do we start to build early patient access to experimental therapy? Part of that seems like right now the reason patients put on very experimental therapy, by the time they are, we know they're tired. Is there a way here to risk identification to tell those patients on
Yeah. Yeah, there's unfortunately a seldom used strategy which is called sort of window of opportunity strategy, where patients before being put on, say, first line are given for a short period of time an experimental drug that seems specifically matched to their sort of genetics, for instance, and you can test whether that will work. If it works, you'll keep them on, otherwise you immediately switch them to first line. I think this is a model that we need to expand on and sort of make it almost the standard, because the reality is that, and I put that point there, forgot to explicitly mention, patients are too sick by the time they are enrolled in clinical studies. This is a major problem because we know that the technology and the science behind cancer drugs today is significantly more advanced than the technology we used to develop cancer drugs 20 years ago, which is what first-line defense is. And yet, we cannot prove that because by the time the patients are put on those new drugs, they're actually extremely sick. So I think this is something, again, from a policy perspective, need to be addressed. And I think there are tools to be able to do that.
Thank you very much, Andrea. We'll have more time for discussion in a few minutes. Our next speaker, I'm very happy to introduce Ellen Feigl, who's going to be talking to us about stem cell therapy. Ellen, of course, all of you know from her many contributions here at the NCI while she was here, is now at the California Institute for Regenerative Medicine. Ellen, have you here.